We're talking about the work of this Friedrich Miescher. The question was what? Give a brief on Friedrich Miescher work in nucleic acid chemistry. Okay, so he did a lot of things that contributed to what nucleic acid that we study today as what DNA, RNA, and the rest. Okay, so let's talk about this man's contribution to this field. All right, and this was an exam question. Give a brief on Friedrich Miescher work in nucleic acid chemistry. So for the overview, we say that what Friedrich Miescher was a pioneering Swiss physician and biologist who made groundbreaking discoveries in what nucleic acid chemistry. Okay, so in 1869, while working in the lab of Felix Hoppe-Seyler at the University of what? Tübingen, Miescher isolated a substance from the nuclei of white blood cells. Okay, so he called this substance nuclein, and this is a substance that right now we know it as what DNA. You get? So what were his key contribution to the field of nucleic acid chemistry? So for the isolation of nucleic acid, like we have said, Miescher was the first to what isolate nucleic acids, identifying them as distinct from what the different proteins we have. Okay. Then he also identified what phosphorus and nitrogen. He discovered that nuclein contained both phosphorus and nitrogen, which was a significant finding as at that time. Okay. Then moving, then protamine discovery. Miescher also identified what protamine, which is a protein associated with what nucleic acids in salmon spermatozoa. Okay. Now, hypothesis of heredity. Although initially unsure of his role, Miescher speculated that the nuclein might be involved in what heredity. Okay, so he just guessed it. Then, Miescher's work laid the foundation for future research into structure and function of DNA. So ultimately, this led to what the understanding of DNA as a carrier of genetic information. All right, so it actually did a lot for the nucleic acid chemistry branch. Okay, so that's it for what Friedrich Miescher work in nucleic acid chemistry.